So I'm going to move up from where Sarah was talking about up into the Pacific Northwest, and I have examples from Oregon and Washington. Next slide. Translational science incorporates aspects of science that go back and forth between management, and with the right moment have a policy window that allows us to inform policy processes. I think the important piece about this to note is that science can inform management and management can help to shape science. And I'm going to be talking about an example of what that looks like today. Next. Traditionally, what's been done with science is that we've done what is often called technology transfer, where science informs management, but there's not the feedback loop where different aspects of management or policy can also then inform and shape the science. And that's the part I want to be talking about more today in my example. Next. So these three pieces come together. This essentially says what was on the previous slide, but next, the sweet spot is right in the middle where the three pieces of these come together. Next. Right there. So what I'm going to be talking about is how different aspects of science were shaped by management and policy in an example. Next. Worldwide, freshwater biodiversity is among the most imperiled. Freshwater support up to 1/3 of our vertebrates on the planet, but their biodiversity is less well understood than for other ecosystems, likely due to their relatively high level of endemic species and cryptic species. Next. Traditionally, across ecosystems, methods used have been based on visual detections to identify and count different taxa, which is not always standardized even outside of eDNA, which is what we're talking today. Our traditional methods sometimes lack standardization, and it can be dependent on both practical and taxonomic expertise of who's in the field and who's able to make those identifications. It's often limited to assessments in freshwater. It's often limited to assessments of adults and often of our species that are listed. And because of the nature of accessing streams, rivers, lakes, and wetlands, often our sampling has been limited to summertime sampling. Next. But many of those aspects have the opportunity to change as eDNA came on the scene in 2007. This shows an eDNA sampler that we use. We build these out of PVC piping, and we set them up so that we can operate different number of replicates that we want for the different projects that we're working on. So this one just shows you of us collecting a 1 liter water sample and pouring it into our filter funnels, and we're able to process, in this case, 4 replicates at one time. Next. So I want to move us into stream environments, into the upper extent of streams. So where streams begin on our landscapes and where fishes and amphibians bound. And this shows an example of what that looks like. This is a private land ownership, and you're able to see leaf trees down the center there where the stream is located. Next. So fish-bearing reaches across Canada and the United States have greater protections than portions of streams without fish. And that's shown here across the different regions. And you see that the blue bars are generally at or higher than the other bars. So when fish are present, the buffer widths and protections in those streams is greater. Next. This is an example from the state of Oregon, showing how different protections at the where fish are found in streams also then vary by land ownership. So I just mentioned that they're greater when fish are present, and you can get an example here of forest lands on the bottom based under the Northwest Forest Plan, and you're able to see the buffer widths there shown in green and fish bearing portion, are greater than there are for state lands, shown there in the blue bars down the center, and our updated current Oregon forest practice rules at the top. And you're able to see the different protections for our private lands and how that varies across the land ownerships. Next. So in addition to the fish boundary being important for forest practices, forest management reasons, it's also important ecologically. And what you're looking at here are each point represents a different location on streams. So we sampled 4 different streams, 8 different locations on those streams longitudinally, moving from downstream to upstream. And where fish drop out in the stream network marks an ecological shift in our freshwater assemblages, with all of those containing fish shown there in blue, and the other ones are where amphibians, different compositions of amphibians are found. Next. And so you're able to get a sense of what that looks like here with the species laid on top of the ones that are present at those sites. Next. So thinking about our traditional approaches and eDNA, I'm going to be talking about how these two compare to each other at the site or the study that I was just presenting. And so we want to understand a little bit more about these different approaches as we are going to move to inform management and policy. We need to, and to set up our different guidelines, we need to understand a little bit more about the different approaches. Next. So what you're looking at here are electrofishing results on the left side and eDNA results on the right side. You're looking at 4 different streams. So each location down the center is a different location. So GS8, Rock 8, those are different stream names. And you're looking at the species down the left side. And so you're able to compare the two by just looking between the 2 different panels for the presence of the different species. So if it's present, it's found there in orange, absent or not detected, it's blank. And if we didn't measure it, it's grayed out. And so what you're able to see here is that we have different detection of different species for the different approaches. Next. Next. And you're able to see that eDNA identifies cryptic sculpid lineages. So the sculpin detections reveal this cryptic species complex. In the field, we were able to identify 2 species, and based on the genetic information that came back with our multi-gene eDNA metabarcoding, we're able to detect 4 to 14 different species or lineages in these different stream environments, which has ramifications for fish conservation, potential pockets of endemic species, and possible identification of new species in these different streams. Next. So thinking of understanding more about the upper extent of fish, we did a study where we wanted to compare electrofishing to eDNA as an approach to identify the upper extent of fish. As many of us know, eDNA does a really good job with identifying species presence. And so what we did is we went out to 60 streams on the west side of Oregon and Washington, and we went to 8 different locations on each of those streams. So you can see an example of one of the sites with the 8 different locations down the center. And then we have different results shown with the eDNA results as an example of one of the streams. You're able to see detections at the first 4 locations, and with electrofishing, you're able to see detections at the first 3 locations. So where there's colored circles, they're detected there, and if it's grayed out, there's no detection. Next. So of those 60 streams, we wanted to compare these different approaches because we were interested in offering a different approach, being eDNA, for managers to identify the upper extent of fish. So at these 60 streams, we first, with our electrofishing, needed to identify which species were there. Next. And so of the 60 streams, 58 of them had coastal cutthroat trout as the uppermost fish. And 2 of the streams had, one had a sculpin and one had a coho juvenile as the uppermost fish. Next. And then again, those 60 streams, we wanted to understand the different approaches. And so we, as I mentioned, we compared eDNA to electrofishing to understand if there was agreement between the approaches or if one of the approaches detected fish above where the other one was. Next. So of those 60 streams, eDNA detected fish at or above where electrofishing was, and or I should say above in over half of the streams. They were in agreement in just under 30% of the streams, and electrofishing identified above eDNA in 20% of the streams. And related to the detection of electrofishing, the way that the sampling protocol works is that we sampled with eDNA first. We went down to the streams and we collected anywhere from 12 to 16 different sites because we didn't know where upper extent of fish was going to be based on electrofishing. And so we had to collect more water than we actually processed. So we collected all these additional samples and sites. And then when the electrofishing results came back in, we set it up so that 3 of the sites would be downstream. 3 of our eDNA sites would be downstream of where electrofishing upper extent ended, and 5 of them would be above it. And so we essentially adjusted our sampling scheme in real time in the field. But just to get back to that electrofishing point, so when we electrofish, again with eDNA, we collected our water samples by walking on the bank and collecting them every 50 meters from where we started in the stream. And our electrofishing protocol is that you're walking in the stream, and so you're moving fish. And so it should be noted that that 20% of electrofishing detecting fish above eDNA, we only detect them 1 habitat section above where we do with eDNA, and it potentially could be because we are walking in the streams, and so we essentially pick the fish upstream in some cases as we electrofish. Next. So a subset of some of the data is shown here where trout eDNA was detected above electrofishing in most of the streams. And as I mentioned before, we sampled at 8 different streams. And so what you're looking at here is like mirrors. And so you can compare what you see on the left side to what you see on the right side, with the site 1 being closest to the center. So they're mirrors of either side. And you're able to get a sense of the sensitivity of eDNA and its detections. And the darker the orange is, the more replicates that the DNA was found in. And so the lighter the orange dot, it was detected in only 1 of our eDNA replicates. And then the different size of the bubble had to do with the strength of that signal. So the bigger bubbles, the strength was stronger than the smaller sized bubbles. Next. So the results from this suggest extending the upper extent of fish boundary beyond the known boundary. So essentially with eDNA, and you're able to see this with just the percentages down each side, you're able to see that by design, the uppermost fish detected by electrofishing fell between sites 3 and 4 with the eDNA. So we always expected to get detections below that, and above that, we weren't necessarily sure what we were going to find. And so our results suggest extending it beyond the known boundary of where fish end. But this is where this piece related to shaping science to inform management come in. I didn't mention this earlier, but there was a lot of time that went into setting up protocols, both in the lab and the field, that were agreed upon across private, state, and federal landowners. So we had to have an agreement in how we are going to sample with our traditional approaches, how we were going to make the calls of what the upper extent of fish was. And we had to have an agreement for how we are going to be sampling for eDNA, how we are going to deal with aspects related to contamination. And I think we talked about potential results, but I think as a lessons learned and going into all projects since, we now beforehand also do agreed upon implications based on potential results. So we lay out scenarios of potential results, and then talk about what that would mean. If we detect eDNA at these sites, what do we all agree the implications of such results would be? Because we did have situation where eDNA was shown to be more sensitive to electrofishing, and not everybody necessarily liked that response. And so that was a lessons learned for us with dealing with that. Next. So this is a different study with the same fish in Oregon, and it's looking at coastal cutthroat trout. So in addition to presence and occupancy, you can get at diversity. And so we're able to identify genetic diversity of coastal cutthroat trout across these different streams where we sampled. And you're able to see that in the coastal watersheds that there's private haplotypes that we're detecting there in one of our coastal river, as well as the Umpqua River. Next. And as a result of this work that we did in Oregon, and because the timing was right, that they were writing up new forest practice rules, written into the new forest practice rules are use of eDNA surveys to determine the upper extent of fish. And this is just a little snippet of where it comes out of the Oregon Private Forest Accord and the language in there related to eDNA. Next. As another example of implications related to eDNA, this has to do with stage 0 restoration. And this, we had to have these agreed upon protocols working with different landowners for how we would sample. And so what you're looking at here are eDNA detections pre-stage 0 restoration. And so what you want to make sure you're noting here is the flow path of the river, and you see that in blue. And then you're able to see our different detections of our different fishes, amphibians. We have some pathogens noted there. We have macroinvertebrates, beaver, and you're able to see what those detections looked like pre-restoration. Next. And then you're able to see the additional water with the new restoration that went in, and essentially the different habitats that were created, and the different species that then, were able to move up and occupy these different habitats. So again, coming back to those agreed upon protocols, it was important because we needed to be able to sample across a landscape that was going to be changing. And we talked about implications of our agreed upon results in this study. Next. So take home messages, just to kind of wrap it all up, is that science can be shaped to inform management. I've shown an example of what that looks like here. And so essentially co-production is about relationships, and that shaping of the science takes time to develop and time to work through the different pieces of what that might be related to trust that's been talked about by some of the other speakers earlier. eDNA is more sensitive than electrofishing at identifying the upper extent of fish, as it is, in many other studies comparing to traditional samples. And we show extensive diversity in watershed using eDNA. Some of it was previously hidden, which changes our understanding of presence of species in these different systems, and then our ability to track, protect, and manage them. Next. With that, there's many people across different organizations and affiliations to thank. So thank you, everybody. And next, with that, I'll take any questions if there's time.